have a cell with its nucleus. The cytoskeleton is a network of interlocking filaments that travel all throughout the cell. These filaments actually define the shape of the cell. Many materials travel across these filaments to be transported from one place to another within the cell. It is actually how the language of the cell itself is spoken. An example of this is the protein that carried the vacuole in Miss Donnelly's video. It traveled across the top of the cytoskeleton. The three protein filaments that make up the cytoskeleton are actin filaments, intermediate filaments, and microtubules. Alterations in the cytoskeleton can cause many diseases, one of which is Parkinson's. Parkinson's occurs when Lewy bodies develop inside of the cytoskeleton. These Lewy bodies are made up of a protein called alpha-synuclein.
Centrioles come in pairs of both a mother and a daughter. They are made of microtubule triplets with distal appendages and subdistal appendages. There are two main functions of centrioles. Their main function is to aid in the cell division of animals. In order to do this, the centrioles create spindle fibers that help in separating chromosomes during mitosis in animals. The centrioles are made of microtubules, which help to complete the 6 parts of cell division. During prophase, the first stage, spindle fibers begin to form from the centrioles and the chromosomes become visible in the center of the cell. In the second stage, the metaphase, chromosomes are seen lining up along the equator of the cell, and sister chromatids are attached to opposite spindle fibers. The next phase, anaphase, is where sister chromatids begin to be pulled toward opposite poles by the spindle fibers. This change causes the cell to be elongated. Next, telophase is when the chromosomes reach opposite poles and decondense as the mitotic spindle breaks down. Cytokinesis is the final stage of cell division. In this stage, the daughter cells are completely separated from each other, each containing identical DNA. The second function of centrioles is to help create cilia and flagella for the new cell, which helps it to be able to move around. A disease called microcephalic osteodysplastic primordial dwarfism type 2 is a direct symptom of defective centrioles. This phenomenon occurs when the protein paracentrin mutates from its normal form.